So I think I've reread this excellent review, if you know this review. So this excellent review published 4 years ago, and those people, maybe you know, wrote in the therapeutic section, they wrote, at present there is no treatment of proven efficacy for cadazil, either for the disease or for the main symptoms. So treatment is thus entirely pragmatic. So, and unfortunately, I don't think that many things have dramatically changed during these past years. However, I do believe that there is room for optimism and hope. So, Marie-Germaine, you were right. I think it's time to think about potential therapeutic avenues, even though there are still many gaps in our understanding of the disease, and at least to answer to Anu's question. So, I won't make a review, an exhaustive review on all the possibilities of how we could imagine to treat cadazil. Cadazil is a chronic disease, and it depends on at which stage, so I won't talk about the possibilities of how we could protect the white matter, how we could promote rehabilitation. Okay. I would like just to focus on what we have, I think, what we believe is important for this disease is that excess of NOD3 ECD maybe is not very good for the brain vasculature. So maybe we can speculate a bit, because I think we are Friday afternoon, and in my lab on Friday afternoon we are allowed to speculate. Only on Friday afternoon, it's true. So I would like to propose to show you 2 concrete examples of how we could reduce the amount of NOD3 ECD deposits if we assume that the accumulation of NOD3 ECD deposits is important for the pathophysiology of this disease. So the rationale is this hypothesis of NOD3 ECD cascade hypothesis, and there are very schematically 3 possible therapeutic interventions. So we can think about reducing the production of NOD3, we can think about preventing the aggregation of NOD3 ECD, and we can think about facilitating or promote the clearance of NOD3 ECD. However, we have to take into account multiple scenarios under this hypothesis. We have to take into account that it could be a NOD3 ECD trigger scenario. I mean, and this is extremely important to define the therapeutic window. If it's a NOD3 ECD trigger scenario, I mean that if the therapeutic comes after the appearance of deposits, okay, that won't work. So if it's a NOD3 ECD threshold scenario, you can imagine that if you intervene before this and below this threshold, it will work. And if you intervene after, that won't work. And the final scenario is if it's in a NOD3 ECD driver scenario, there is some hope that the therapeutic could work at multiple stages. And finally, you have to think about the NOD3 ECD irrelevant scenario. So how can we, what kind of possibilities you have to reduce NOD3 ECD production or prevent aggregation or facilitate clearance? I don't have any magic bullet, but I would like to show you 2 concrete examples for applied and in clinical trials for neurodegenerative disease. So the first one is the antisense oligonucleotide approach, and which could be applied to reduce the concentration of a toxic, especially of a toxic protein. So how does it work? I'm absolutely not an expert in this field, so what I've read, exactly as Joanna said, I can only tell you what I have understood from the literature. So how does it work? These are very short synthetic chemically modified nucleic acid that binds to the RNA, so the RNA makes the protein, and you can by binding these short oligonucleotides which bind to the RNA, trigger cleavage because it forms a duplex, and it activates RNase H cleavage and promotes the degradation of RNA. So this is the gene silencing mediated through degradation mechanisms. So there are other ways to impact the RNA and at the level of metabolisms. So the nucleotide can bind and modify splicing. So very schematically, there is a bunch of ASO which works through the degradation process and other ASO. And you may think about the Duchenne dystrophy, which modulate the splicing of a gene and lead, in this case, to the production of a protein which is modified because exons have been removed, and to produce a shorter protein. So in the first strategy, you completely degrade the mRNA, and in the second, with the second strategy, you produce a shorter protein or at least a different protein. So you can have a non-selective approach. I mean that you can target both the wild type and the mutant alleles, and in this case you will reduce the production of both the wild type protein and the mutant protein. Or you can have, using a molecular biology technique, you can silence, but this is more complicated, and it may also be difficult because it will have to be specific to each patient. It can be an allele-specific silencing. So this approach is very interesting and is already in the clinical trials. So the ASO, these short oligonucleotides, have very favorable characteristic because they have long tissue half-life, and this has been controlled not only in the mouse, but also in humans, and the half-life is about 1 month. If you infuse into the CSF through the implantation of a mini pump and with an intrathecal infusion, there is a very efficient delivery in the CNS, and it's very efficiently uptake in neurons and glia. But to the best of my knowledge, I think there is not any paper looking at the smooth muscle cells. In addition, it can enter very easily into the nucleus, and because we are using naked ASOs, the adverse effects are very minor, and you can use very precise dosing. So very interestingly, there have been several reports of very successful preclinical studies in Huntington's, in mouse models of Huntington's disease, but also in non-human primate models of Huntington disease, and also in ALS. What is interesting and intriguing for me is that the beneficial effect may persist for longer than target suppression. I mean, this is the holiday effect. I mean that the people who have done these studies have tried a transient infusion, and they stopped the infusion, and they saw a beneficial effect several months after the interruption of the infusion of the ASO. So a reason to have some hope is that a recent paper has been published and showing a well tolerance in man, and this is a phase 1 study in patients, so having a genetic form of ALS due to mutation in SOD1 gene, and there are some ASO in phase 2 in amyloidosis caused by transthyretin mutation, and there are phase 3 studies, clinical trials for the Duchenne disease. So it's not so this therapy is not for the next century, it's for the next years. So can we think about ASO for CADASIL? I think so, because disease with a toxic gain of function, the disease with a toxic gain of function mechanism, as we suppose is to occur in CADASIL, are very good candidate for such therapeutics. So the idea is to reduce the expression of NAR3CD, and in this case, you will reduce all the production of all NAR3 protein. So I won't go into the details, but at this point, we do not know if these are the multimers or oligomers or monomers of NAR3CD which are important for the disease. So with this strategy, we can reduce all these kinds of species. So we can, and this is to point out that it's extremely important to continue and to pursue research and research of mechanisms. For example, it's very important to know if there is a contribution of a loss of function in cadavers, because if the reduction of NAR3 does not absolutely contribute to the disease phenotype, as our data strongly suggest, you can consider a non-selective silencing which will down-regulate both the mutant allele and both the mutant and the wild-type allele. However, you have to think about the potential adverse effects of reducing the wild-type protein at the level of the microcirculation and in the periphery. So I already discussed the problem with smooth muscle cells uptake, but I think there is no reason to think that the smooth muscle cells are very special and won't be permissive for this ASO. And so we are not, at this point, I think we are not ready to start such studies because we need some in vitro assays and some humanized mouse models to screen and to test ASO. However, I think that iPS cells, because at this point we don't have any in vitro model which recapitulates the accumulation of the extracellular deposits. So iPS cells, if we can isolate iPS cells and prepare iPS cells from patients and recapitulate, and if these iPS cells can recapitulate the NAR3CD deposits, we could screen this strategy. And there is also a key question, by how much should NAR3CD production be lowered? So we need more studies, and we need more studies before embarking into this strategy, because it's very important to determine the therapeutic and to determine the therapeutic window. So the second approach we can think about is vaccinotherapy. Okay, you can, I'm sure many of you have in mind vaccinotherapy does not work. I will show you that it's a bit more complicated. So for vaccinotherapy, I think we can take advantage of 14 years of experience in Alzheimer's disease. So this vaccinotherapy approach, for those who are not familiar with vaccinotherapy, is based on the amyloid cascade hypothesis. So this hypothesis which posits that the accumulation of A-beta oligomers, soluble oligomers of fibrillar species triggers this cascade of events which will lead to neurodegeneration and vascular lesions. So there are two types of vaccinotherapies, either passive approach, so you inject the antibodies, or active immunization. And with this approach, you inject the peptide, and this is a patient who will produce the antibodies. So in mouse models, it works. It definitely works. It attenuates A-beta deposits. In patients, the pathological studies have shown that it works. It reduces the amount of deposits. But you may think that how, with the normal blood-brain barrier, how the antibody can enter into the CNS. And there is a huge experience with the labs who have worked and we have used this approach, that even though, even with a preserved BBB, there is a small amount of antibody which can enter into the brain, and because the antibody recirculate between the CSF and the plasma, so the small, at each passage, a very small amount of antibody enter. So it has been calculated that about 0.05 to 0.2% of the antibody can enter and can have access to the brain. So the mechanisms of passive immunizations, I should say, are multiple, but I think controversial, and not very well known. So it has been suggested that there was a peripheral sink mechanism suggesting that the antibody titrates in periphery, the A-beta. It has been suggested that it could be phagocytosis by microglia, inhibition of aggregates. But if you take each of these mechanisms one by one, you can argue against. So to be honest, we do not know exactly how it works. So, and we have to be aware of a number of adverse effects, but I would like to clarify some issues regarding the adverse effects. So, one of these adverse effects is the aseptic meningoencephalitis, but this adverse effect has been observed with active immunization. So, you probably heard about these two adverse effects, which are first the occurrence of edema, vasogenic edema, and exacerbation of amyloid angiopathy, and which can trigger some hemorrhage and some rupture of vessels. However, a recent study which has been published in the Lancet Neurology last year has reanalyzed these adverse effects, because you can see the consequences of these adverse effects in humans treated by bapineuzumab, and they reviewed the MRI. And the take-home message is that in 70% of the cases, there were no symptoms. So these lesions, so the abnormalities seen on the MRI were completely asymptomatic. And in addition, this effect in some patients, the immunization has not been interrupted, and these adverse effects were not exacerbated by, despite the passive immunization. So we have strong suspicion now that these adverse effects, which correspond to the mobilization of A-beta around the vessels, and which trigger transiently this inflammatory reaction that exacerbates the amyloid angiopathy, is a transient adverse effect in both mouse models and in patients. So, could we use this approach in CADASIL patients? No. This slide is extremely important. So, you may have in mind that the vaccinotherapy does not work in Alzheimer's disease, but I should say that the clinical trials, there are many reasons to explain the failure of these clinical trials. And the first one is that these clinical trials have been given too late. The experimental studies in the mouse have shown that it worked at the very beginning, and the vaccinotherapy has been tested in patients at the end stage of a disease. So, showing that there was a complete, absolutely no alignment between the preclinical studies and the clinical studies. And I think it's a major reason for failure. A second reason, it's the heterogeneity of patients which have received this therapy. You can see that 30% of patients did not have any amyloid deposits. So, and in addition, in this patient, we can think about the mixed nature of dementia. So maybe we do not see any effect because maybe we can postulate or speculate that we treated the amyloid effect and we did not treat, for example, the vascular effect. So just to give some hope for the vaccinotherapy, and I think we are doing now what should have been done first. So there are ongoing prevention studies and targeting pre-symptomatic mutation carrier. So pre-symptomatic, so at the early stage of a disease, and mutation carrier, so and a very homogeneous population, and we are sure that these patients have the disease. So there are 3 ongoing prevention studies. So this one targeting a very large Colombian family. A second prevention study targeting pre-symptomatic carriers carrying a mutation in PS1, PS2, or APP, and a large prevention study targeting cognitively normal subjects, but identified on the basis of PET scan showing amyloid deposits. And we have to improve the design of the trials. So can we think about vaccine therapy in CADASIL? I think so. So first, the deposits are extracellular, and the main difference with Alzheimer's disease is that we have a possibility to monitor the deposits of nitric-CD in live mice and patients, because there is an accumulation in periphery. And third, I think it's a very beautiful proof of concept study of a nitric-CD cascade hypothesis, because if we can prove that by clearing these deposits we can rescue or prevent the disease, this is a very nice proof of concept study. However, we have to be aware that there are some similarities with the Alzheimer's disease, but many, many differences. There is no reason to think that the nitric-CD is soluble either in the CSF and the plasma, but to be very honest, we never tested this. Second, we can think that by analogy with the Alzheimer's disease, we can think that if we vaccinate, we could exacerbate rather than decrease the deposits, and we could exacerbate the deposits around the vessels and promote vessel wall inflammation upon treatment. And we have absolutely no idea of the efficiency of clearance of deposits around the vessels. And also we have to think about the potential deleterious effect on the wild type nitric function, and the same comment for the various therapeutic window, which depends on various scenarios. So Mark, can you comment on the next slide, boosting lymphatic clearance?
Okay. Thanks, Anne. I thought she was only going to give me one slide. I guess she gave me two. This is, I think you heard about earlier today, this is a relatively, I guess, old concept, but newly presented, and with some new evidence from Mike and Niedergaard's lab for clearance of all the waste in the brain, called lymphatics. And so this may be, if there's a possibility of increasing clearance, if again it's soluble, this may be another mechanism to clear the extracellular, the Notch3 extracellular domain or deposits out of the brain. This is, I guess, still a sort of speculation level. This is a slide we started making, and this gets to another fundamental issue. Is the defects in cerebral vascular function, how much do they contribute to the pathology? And I gave you two experimental examples, one from Anne's lab with TIMP3 genetic reduction, one from ours of applying EGF to reduce the levels of Kv1.5 channel. So we restored at least a myogenic tone. The question is, we could do that and at least start with the animal model, and does that affect the pathology? Don't know, but certainly something that happens early in the lifespan of the disorder. And then I'm hearing the really intriguing data about the two other families that have Kv1.5 mutations, combined with, as I mentioned, experimental evidence from Masayo and George Wellman in Vermont that at least in animal models of subarachnoid hemorrhage, it involves the downregulation, maybe through the same system, of Kv1.5, suggesting maybe common threads amongst a number of cerebral vascular disorders, and that modulating them could, and restoring, at least to some degree, normal feedback in cerebral vascular function may improve the disease prognosis or slow it down, slow down the disease progression. This is a fascinating area. I know Costantina's talked about this as well, is that, how are these, sort of small building cerebral vascular disorders contribute to cognitive decline later in life? It doesn't take a lot to think if you have a dysregulation of local blood flow. Unfortunately, the pharmacology of these potassium channels, although a lot of drugs for ion channels, is not good. We have non-selective 4-aminopyridine as a blocker, which we would want an opener, and these have been rather resistant to development of openers, unlike some other types of potassium channels. And the other problems, they're expressed throughout the body, including the rest of the vasculature. And I had dealt with long ago looking at openers of ATP-sensitive potassium channels. First, they were looked at for lowering blood pressure. Monoxidil sulfate, in fact, works this way. They cause enormous reflex tachycardia. They're actually very lethal hypotensive agents. They weren't controlled. Then they looked at it for urinary incontinence. Again, you had systemic effects. So there's been a long history of trying to deal with potassium channel pharmacology, and some things work, some don't. But the ideal thing would be to, if we could restore and test the hypothesis that restoring KV channel function would restore, would not decrease, perhaps contribute to, we don't know, the white matter lesions, lacunar infarcts, the progressive cognitive decline. But this applies to a lot of cerebral vascular disease. And one thought, and we talked about this, and Anne brought this up, in the advantage of being able to, the gum deposits and the Notch3 mutant Notch3 mutant forms are expressed in the vasculature throughout the body, and it's typically assayed by peripheral arteries or skin biopsy. Well, maybe we're looking in the wrong place. The question is, why isn't the heart? Why isn't the kidney affected? It has autoregulation. It has blood vessels, it has Notch3, extracellular domain, gum deposits. Maybe it's something about the brain, and one obvious thing which we're looking at is one big difference is surrounded by astrocytes. And so it might be something about the astrocytic end foot and one of the proteins there that's causing the pathology. And obviously it's not causing, anyway, overt renal or heart problems. So maybe we're just looking in the wrong spot. Maybe we have to go across the smooth muscle and jump that gap to the astrocytic end foot. And maybe would have then another candidate for, antisense or immunotherapy. So
I have 2 more slides. And we'll finish. So definitely if we move on preclinical studies, we need to be very rigorous and not to do the mistakes which have been done, and especially to control for internal validity, construct validity, and external validity. There are many, many, many reports in the literature where preclinical studies have been done on various samples of mice with unblinded evaluation without any monitoring of the health status. And we know that the health status is very important, without any control of gender, of genetic background of the mouse, with any randomization. So we need to be as rigorous as clinicians. And to finish, and this will be my last slide, I think, this is my opinion, that Cadazil is a very good candidate disease for ISO or immunotherapy. However, the optimal treatment, we have to be aware that such treatment maybe will be efficient at the pre-symptomatic stage. So we have to think very differently. So, and the optimal treatment will probably be a combination of treatments. So there is a big need to pursue research on the mechanisms. And it's a very obvious things, but very important. We should not advance into human testing preclinical candidates that have not been very rigorously demonstrated. There is so many failures of drugs which worked on preclinical studies and which do not work. And the pharmacies are absolutely not very keen to continue to support research if we are not able to demonstrate that candidates which work in preclinical studies can have some effect in patients. So it's extremely important to align the design of the preclinical studies with human clinical trials. And I think it has been the very big mistake with vaccinotherapy in Alzheimer's disease. And for the clinicians, if we are embarking on preventive trials, we absolutely need reliable outcome measures at the very beginning and at the pre and early pre-symptomatic stage. So you have a lot of work to do also. Thank you.